The Gaussian distribution, also known as the normal distribution, is a fundamental concept in biological experimentation, describing how many natural biological variables tend to cluster around a central value with symmetric variation. Understanding this distribution is essential for analysing experimental data, as it forms the basis for statistical tests, confidence intervals, and error analysis. In this lecture, we will explore what the Gaussian distribution is and how to test experimental data for normality or Gaussian behaviour. Biological data shows a lot of variability, so experiments are often looking for small changes in the face of large amounts of variation, and statistical tests are required to assess whether observed changes are real or just part of the normal variation. The Gaussian distribution results from many random and independent factors acting in an additive manner to affect the data recorded. It is important because many of the widely used statistical tests were developed on the assumption that data vary according to the Gaussian distribution. These tests include t-tests, ANOVA, and regression analysis. Because these tests assume Gaussian behaviour, they are not appropriate for data that follow different distributions. The main data distributions encountered in physiological and pharmacological experiments are the Gaussian or normal distribution, the log normal distribution, and the exponential distribution. Examples of data following Gaussian behaviour are the heights and weights of human populations, resting blood pressure, equilibrium plasma concentrations of drugs, and tissue responses to drugs such as vasodilation. The distributions of these parameters follow a bell shape with symmetry around a central point. Bacterial and viral growth rates, drug elimination half-life, blood hormone concentrations, and reaction times all follow a log normal distribution. This looks like a normal distribution, but it is asymmetric with data skewed to the right because there are more small values and few large ones. An exponential distribution is seen for the open and shut times of ion channels, neuronal firing rates, the lifespan of cells, and the time between heartbeats at rest. This is a right-skewed distribution with many small values and a steady decline in the number of observations as the values get larger. The last two distributions will not be covered here. Instead, I will focus on the Gaussian distribution and cover the other distributions in a future video. The simplest way to illustrate a Gaussian or normal distribution is with an experimental example. A class of 100 students was interested to know how much alcohol they would need to drink to reach the legal limit for driving. So they each drank a glass of vodka, making sure everyone had the same amount. After 1 hour, a blood sample was taken from each student and the concentration of alcohol in their blood was measured. They observed a lot of variability in their blood alcohol levels because individuals differ in how they metabolize, absorb, distribute, and eliminate drugs like alcohol. To investigate the variation, the students drew up a table listing a series of possible concentration ranges and the number of students whose blood alcohol fell within each range. On a graph, they plotted the number of students on the y-axis against the midpoint of each concentration range on the x-axis. This showed that the relationship followed a bell-shaped curve. The blood alcohol concentrations of most students clustered around the middle of the curve, with very few at the extreme ends. The curve was symmetrical around the midpoint. You can see this by tracing along one side of the curve, flipping the trace and superimposing it on the other side. This symmetrical bell-shaped curve is known as the Gaussian or normal distribution. The centre of the distribution is the mean, usually indicated by the Greek letter mu. In this example, the mean blood alcohol concentration was 0.5 milligram per mil, which happens to be the legal limit for driving in many countries. What it demonstrates is that due to individual variability, for 100 people drinking the same amount of alcohol, half would pass a breathalyzer test an hour later, but 50 would not. If you were to repeat the experiment with a more select group of students, restricted, say, to athletic young males of the same age, height and weight, you might expect to see less variability in their blood alcohol levels. The data would still show a Gaussian distribution with the same mean, but there would be tighter clustering around the mid-value and a narrower spread of values. In biological experiments, it is important to know how spread out the data are. The measure that tells us this is the standard deviation, usually indicated by the Greek symbol sigma. It's calculated using this formula. The standard deviation can easily be obtained using statistical software packages and Excel spreadsheets. However, it is important to understand what it measures, so I will talk you through the equation. The first step is to calculate the difference between each of the individual measured values and the calculated mean value. In other words, how much each value deviates from the mean. Each difference is then squared and all the squares added together. Apart from being mathematically correct, the differences are squared because some values are larger than the mean and give a negative difference, which would cancel out positives. It also gives more weight to larger deviations. The total is divided by the number of observations to find the average difference, and finally the square root takes you back to the original units and gives you the standard deviation. When marked on the Gaussian distribution, the standard deviation is shown twice, once on either side of the mean value, representing plus or minus 1 standard deviation. An empirical rule is that 68% of the experimental values fall within 1 standard deviation of the mean, and 95% of values lie within 2 standard deviations of the mean. Our original example employed 100 students, but biological experiments rarely collect that many observations. Let's see what happens if we reduce the cohort to only 20 random students. There are many fewer values in each blood alcohol concentration range. When the data are plotted, they still follow a Gaussian distribution. And if we scale up the plot, you can see that it is the same Gaussian distribution as before, with the same mean and standard deviation. Let's reduce the cohort even more, to 10. This is still quite generous compared to the numbers of repeats often employed in biological research. The number of students achieving blood alcohol concentrations in each of the ranges is now very small. A Gaussian distribution can still be discerned, but the fit to the data is not as good. With even smaller cohorts, there would be too few data points to plot a frequency histogram like this and check for a Gaussian distribution. You need a large amount of data to test for Gaussian behaviour in this way. Another way to test if a distribution is Gaussian is to construct a quantile-quantile or QQ plot. A quantile is a value that divides a probability distribution into equally sized intervals. And a QQ plot compares the quantiles from experimental data with theoretical quantiles from the normal distribution. So, what does that involve? The theoretical distribution is centred around a mean equal to 0 with standard deviation equal to 1 and plotted as the probability of a value being recorded experimentally. The distribution can be divided into 6 equal quantiles, from -3 to +3, a quantile here being equivalent to 1 standard deviation. The values on the x-axis are the z-score and tell you how many standard deviations a value is from the mean. The cumulative probability of a value being less than or equal to the z-score has been worked out for the distribution and published in a standard reference table. It is known as the Standard Normal Probability Distribution Table. This section of the table covers the first half of the curve from a z-score of -3 to 0. To read the table, locate the first two digits of the z-score in the left column and the third digit in the top row. For example, find the row at -0.2 and the column at 0.05, making up z equals -0.25. The value where they intersect is the probability of recording values smaller than or equal to that z-score. To explain how a QQ plot is constructed, I will walk you through the process with experimental data. In this study, volunteers were administered a new drug, and the concentration of the drug in their blood was measured 1 hour later. These are the measurements obtained from each volunteer. The values are entered into a table, ranked in order from the lowest to the highest value. Each rank is assigned a percentile value, calculated by dividing the rank -0.5 by the total number of data points. These values are then used to find the theoretical quantiles, or z-scores. This is the mathematical representation of the process. The Greek letter phi represents the cumulative distribution function, giving the probability of a variable being less than a given value. The inverse of the function gives the z-score at a given probability level. It looks complex, but all it entails is reading the table that you saw earlier in reverse. This time, locate the probability values that match or are closest to the percentile ranks in the table. The yellow highlights show the first three values from our data table. Now read off the corresponding z-score by identifying the first two digits in the associated left-hand column and the third digit in the top row. The first data point gives z equals -1.38. Repeat for the other percentile ranks. To make things easier, the inverse cumulative distribution function is available in Excel, allowing you to quickly find any theoretical quantile. Once you have entered the percentile ranks in a spreadsheet, click on an empty cell and type the equal sign to bring up the choice of functions. Select Norm SINV by double-clicking, then click on a percentile rank value to select it as the argument for the function. Entering the formula will return the theoretical z-score for that percentile rank. Copy and paste the cell to apply the formula to all values in the series. Now construct a graph with the theoretical quantiles on the x-axis and the original data on the y-axis. This is the QQ plot. If the data are normally distributed, the relationship between the data and the theoretical quantiles will closely follow a straight line. Deviations from a straight line suggest skewness or non-normality in the distribution. Statistical and graphing software packages often have an option to create a QQ plot. And as you can see, it is a good way to check for normality where the sample size is small, only 6 in this example. Statistical and graphing software offer several statistical tests that can be applied to assess whether data are normally distributed or not. Each test examines a different way in which the data might deviate from Gaussian behaviour. I won't go into the details, but will give an overview of what they do. Beginning with the Kolmogorov-Smirnov test. It works by by comparing the cumulative distribution of the actual data to the cumulative distribution predicted by a true Gaussian distribution. This test is quite insensitive and only really works with large sets of data. The Anderson-Darling test is an improved version of the Kolmogorov-Smirnov test, which takes more parameters into account, giving more weight to the extremes of the distribution. It works best with small to medium-size datasets and not with sample sizes less than 20. The D'Agostino-Pearson omnibus test calculates the skewness and kurtosis of the dataset and compares these with what is expected for a Gaussian distribution. Skewness is a measure of how symmetrical the data samples are, whether they are centred around a mean or skewed towards one end or the other. Kurtosis is a measure of how sharp the peak of the distribution is. The kurtosis of a Gaussian distribution is close to 3, with higher values indicating a sharper peak and lower values a broader peak. The test produces a statistic that incorporates assessment of both factors. This is the best test if you have at least 30 samples, but it is not applicable to smaller sample numbers. The Shapiro-Wilk test is the most straightforward. It compares the actual data distribution to an ideal Gaussian distribution and checks how well they match. It works with small data sets and is the most suitable if you have less than 30 samples. Each test returns a probability or p-value. A p-value smaller than 0.05 indicates a less than 5% chance that the dataset follows a Gaussian distribution, so it is unlikely to be normally distributed. If p is greater than 0.05, the data likely does follow a Gaussian distribution. It is good practice to test how data are distributed before selecting statistical tests for comparing 2 or more experimental datasets. However, it is important to realise that normality tests give you the likelihood of data following a Gaussian distribution, but they are not a guarantee. Researchers should be mindful of the limitations of tests and their potential pitfalls. Limitations can include small or large sample sizes, or outliers, which are data points that are a long way from the mean of the sample. A small sample size can make it hard for a test to detect deviation from normality. For example, with only 5 points, a skewed distribution may well appear symmetrical. At the other extreme, a large dataset could contain some small deviations from normality that could impact the calculation of p-values while not actually being important. The same problem is seen with outliers, which can make an otherwise Gaussian dataset appear non-Gaussian. An additional issue is that normality tests only check one aspect of the distribution. For that reason, it is wise to combine tests that look at different criteria and use additional methods of visualizing the distribution. Finally, the tests don't tell you if normality matters. Some statistical comparison tests don't rely on a Gaussian distribution, and even t-tests and ANOVA are robust if the deviations from normality are small. If the dataset is small and you are unsure of the distribution, you may be better to use comparison tests that are not specific for normally distributed data. These are known as non-parametric tests. So far, we have looked at the distribution of a single set of data, but experiments compare sets of data, and each set is subject to variation. Now let's consider an experiment that compares 2 variables. So 2 vasodilator drugs, drug A and drug B, were compared on an isolated artery in an organ bath setup. A single concentration of drug was applied and repeated for each drug on 30 different artery preparations. A frequency histogram was constructed by plotting the percent vasodilation on the x-axis and the number of times that vasodilation was recorded on the y-axis. Plotting the data for drug A shows a Gaussian distribution. plotting the data for drug B on the same plot also shows a Gaussian distribution, but it is shifted to the right. That is because the mean percent vasodilation evoked by drug B was larger than drug A. Variability was the same in both groups as indicated by similar standard deviations. The difference between the two data sets in this case is clear, but that is not always the case. Often the distributions are poorly separated, with the mean values close together and standard deviations that overlap. This is an example of what I mentioned at the start: a small difference in the face of relatively large amounts of variability. With small data sets, it would be difficult to determine if the vasodilation evoked by drug B was truly larger than that evoked by drug A. It is much easier to tell the difference between two distributions when they have widely spaced means and small standard deviations.